This is one more type of treatment assignment, blocking. Blocking can be done in both. It can be done during randomization or it can be done during enrollment as well. That is the reason we have termed this as treatment assignment, blocking treatment assignment, not either randomization or enrollment. So that you have to keep in mind. Blocking can be done both for enrollment and both for randomization. So enrollment is simple. When the investigator is given a choice to select the treatment group, that becomes enrollment. When the investigator is given a choice, that is enrollment. When it is due to the chance or it is assigned by the system, then it is called randomization. So yeah, blocking is possible, both type of treatment assignment, that is randomization and enrollment. Now we will look what is blocking. So previously you remember that when we use a simple sequential randomization, so treatment group 1, treatment group 2, so they can be imbalance in the treatment ratio. For example, I want treatment ratio of 1:1, and first subject has come, it has come here, second subject it has come here, third subject here, fourth subject here, fifth subject here, sixth subject here. So what is the treatment ratio here? 2:1, which I don't want. I want to remove this treatment imbalance or imbalance in the treatment ratio. When the treatment ratio is not met, I don't want such type of defect in my system. To remove this, the scientist has come up with the concept of blocking. So what is block? We will look into it. What is the advantages of block? We will look into it. Now take that. I'm arranging the patients into a simple block. I'm taking a block. Okay, this is one block. This block has 4 entries: 1, 2, 3, 4. Okay, this is a block. Think that we will go to multiple blocks first. So 4, 5, 6, 7, 8, 1, 2, 3, 4, 5, 6, 7, 1, 2, 3, 4, 5, 6, 7, 8. Okay, there are 8 blocks here. So in that blocks, each entry treatment group 1, think that there is treatment group 1 and treatment group 2. I want to achieve the ratio of 1:1. What I do is, I will keep this entry for TG1, this entry for TG2, this entry for TG1, this entry for TG1, this entry for TG1. How many TG1? 1, 2, 3, 4. Okay, and this way I keep for TG2, TG2, TG2. Okay, there are 1, 2, 3, 4 TG1s and 4 TG1. So when the first subject comes into the clinical trial, so I will assign first subject to this. When second subject comes, I will assign second subject to this entry in the block. When third subject comes, this. Fourth subject comes, this. So at the end of when the block is completely filled, okay, you can see there are there are 4 subjects in treatment group 1 and there are 4 subjects in treatment group 2, and the ratio is 1:1. Okay, now I have updated the simple sequential randomization with blocks, so that the main part of the block, the importance of the block is to have the desired treatment ratio. It is not only 1:1. If you in some clinical trials they want to have the treatment ratio of 1:2, it can be achieved through block. They can be 1:1:2, 2:2, 2:3. So if there are multiple treatment arms, so this can be achieved through this blocking. Now we will go to single block and multiple blocks. Here you can see this is only one block. We are discussing about single block. What are the advantages of single block? If there is what are the disadvantages of single block? Then only we will go to multiple blocks, right? Otherwise I will not go to multiple blocks. This is fine. I am enrolling the patients, I am achieving the treatment ratio of desired treatment ratio. Why do we need multiple blocks? Okay, we will come up with a special scenario where single block fails. That scenario is, For example, there is a block, this block, single block I have taken. This is the entry 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Okay, there are 14 entries. Same, we will take treatment group 1 and treatment group 2. I want to achieve the ratio of 1:1. We will check. So 1, 1, 1, 1, 1, 1. Okay, 1, 2, 3, 4, 5, 6, 7. This is 2, 2, 2, 2, 2, 2, 2. 1, 2, 3, 4, 5, 6, 7. Okay. This is for treatment group 1, TG1, and these are for treatment group 2. Overall, I have 7:7, right? So my treatment ratio is 1:1. Suppose one subject, first subject 1 has come. I will assign him to here. Second subject has come. I will assign him here. Third subject has come. I will assign here. Fourth subject has come. I will assign him here. Okay. Again, fifth subject has come. I will assign here. Sixth subject, I will assign here. Seventh here. Eighth here. Ninth here. Okay. 10th here. What happens suddenly is the 11th. What happens suddenly is there. How many total patients I need to get? 14 subjects, right? But if I get only I may while randomization, if I am able to get only 11 subjects here, ratio fails. So this becomes 4:7. So I am not achieving the treatment ratio. If there are incompletely filled blocks, if I do it in a single way, so if there is incompletely filled block, what happens is this is a problem. If the block is completely filled in a clinical trial, this is just an example where we have taken 14 patients. Okay, I have seen a clinical trial. There are in a clinical trial there are 24,000 patients. I have seen the clinical trial. I worked on it. So if that is the mega trial and if you have treatment imbalance, then the results will be false. How do we remove this problem, or how do we remove this issue? There comes the multiple blocks. So how multiple blocks can help in this to address this issue, we will check now. So then to look into the multiple blocks issue, so we will again take treatment group 1, treatment group 2. I want to achieve the ratio of 1:1. Now instead of having single block, I will have multiple blocks now. 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3, 4. Now what I will do is I will have multiple blocks. This is block 1. This is block 2, block 3, block 4. Okay, I will have multiple blocks. I will ensure that the block is as small as possible. There is a formula. How do we calculate the block size? So here block size is 4 because there are only 4 entries in the block. 1, 2, 3, 4. So the block size here is 4. First one is, now you have addressed what is block size, right? Right. Now first patient comes, go to this block. For example, okay, this is treatment group 1, this is treatment group 1, this is treatment group 2, treatment group 2. When the first subject comes, you go to this block. Second subject comes, you go to this block. Third subject, go to this block. Fourth subject, go to this block. At the end of the block, every block, treatment ratio is maintained. Okay, if I divide the bigger block into chunks, maybe there will be some thousands of blocks. At the finally, only one block or two blocks are incomplete, so it doesn't make much difference, right? We are almost there to maintain 1:1. It is not 100%, but 99.99% we will achieve 1:1. If there is the last block will be incompletely filled sometimes. For example, there are 900, there are 1000 blocks, okay, but in a trial only 999 blocks are filled, or it can be only 100 blocks are filled, even 100 blocks or even 99.999 blocks. So the error here or the discrepancy here I get is very less. That's the reason we go with multiple blocks rather than a single block. So there is a formula to calculate what should be the block size. We will look into that later. So that is an advanced concept. So depending on the treatment groups, how many treatment groups, how many treatment ratios. So they can be 1:2:3:4. There can be multiple treatment group 1, treatment group 2, treatment group 3, treatment group. So what is the block size? Suggested block size, there is a formula. We will look into that later. So now we understood what is what are the disadvantages of single block, why we are going with multiple blocks. You also know what is block size. Now we will look into block sequence. Block sequence. So some of the statisticians wants to assign this block in a sequence. For example, it is a statistician's preference. Every clinical study will have a statistician. Different statisticians have different opinions, and we have to work with them, and we have to give a system to them to their whims and wishes. So whatever they want, we have to provide in that way. So some of the statisticians, what they want is they want to introduce block sequence. Think this is block 1, block 2, block 3, block 4, and block 5. They will say it should be filled in the block sequence. What they will do is, normally what is the block sequence? This will be the block 1, this is block 2, this is block 3, block 4, block 5. Okay. When first patient comes, he goes into this block. After this block is filled, next it goes into this. Okay. This can be changed also. How this can be changed? Now I am making the blocks: block 1, block 2, block 3, block 4, block 5. Now I have given the block sequence. This has 1, this has 2, this has 3, this has 4, this has 5. Now what should happen? After first patient comes, he goes to this block. Second patient, third patient, fourth patient. Fifth patient comes, it goes to this block. Okay. Eighth patient comes, then fourth block, then fifth block. So it should be filled in the block sequence. The block should be filled in the sequence. That is known as block sequence. Okay. And there is block number. Then what is block number? Block number is nothing but the serial number. It doesn't have any impact, but we give the number in order to know the how many block kit list by looking the randomization list or enrollment list. We have to estimate how many blocks are there. That's the reason it's just a number. It doesn't have any importance. But when the blocks block sequence is not given, so it has to follow the block serial or block number. So that we will discuss in the treatment group pickup. So how the system should pick the treatment group, there we will discuss. One more concept regarding the multiple blocks. Here we have seen there is a block 4, right? 4 entries. There are some statisticians who want the blocks of multiple sizes. Here I have taken the block of 4. In the same treatment list, I can have the block of 2. In the same treatment list, I can have the block of 6, 3, 4, 5, 6. See, 1, 2, 3, 4, 5. think that this is 6. So here at the end of the block 1:1 is maintained, here also 1:1 is maintained, here also 1:1 maintained. So there can be multiple size block sizes within the treatment list. In a particular study, it is up to the statistician. They want to go with single block size or multiple block sizes. The system should support it. So this is also one of the combinations possible for blocking multiple blocks.
ClinGenius Research.